Hey everyone, and welcome back to the deep dive. I know lots of you are prepping for biochem exams, so we're diving into some really fundamental concepts.
Definitely the cornerstones.
Yeah, for sure. And today we're focusing on the roles of water, acids, bases, and buffers in the human body.
Can't live without them.
Absolutely. And to help us with all this, I've got my awesome colleague here, an expert in all things biochemistry.
Glad to be here.
I know you always tell your students that understanding these core concepts is like building a house.
Yeah, like a solid foundation.
Right. Without it, everything else falls apart.
Crumbles.
Exactly. So let's get started with water. You know, that molecule that makes life possible.
Oh, yeah.
That universal solvent, right?
For good reason.
I know. About 60% of the human body is water. And it's vital for pretty much every physiological process.
Pretty much. And it's not just the volume, it's about how it's organized within the body.
Right.
We've got those distinct fluid compartments, like intracellular fluid, which is inside our cells.
Right.
And then extracellular, including interstitial fluid, blood, and lymph.
You know, understanding how these compartments work and how water moves between them, it's critical for everything from blood pressure to nutrient delivery. Right?
Yeah, for sure.
Now, I know from teaching this that students often get tripped up on the concept of osmolality.
Yeah, it can be a bit tricky.
Can you break that down for us?
Yeah, so osmolality, it's basically a measure of solute concentration.
Okay.
Like if you have a high concentration of solutes in a solution, it's got high osmolality.
Great.
Water always wants to move from an area of low osmolality to an area of high osmolality.
To try to even things out.
Exactly. That's the driving force behind osmosis.
So that's why athletes need to replenish electrolytes when they sweat.
Yeah.
They're not just losing water.
Right.
They're losing those crucial solutes too.
Absolutely. It's all about maintaining that balance. Right. And clinically, understanding osmolality, it's like so important for managing things like dehydration and edema.
Right.
You know, for instance, in diabetic ketoacidosis, the high blood glucose levels, that creates a hyperized molar state, and that pulls water out of the cells.
Oh, wow.
Leading to severe dehydration.
Okay, so we've covered the importance of water, osmolality. Now let's get into acids and bases.
The dynamic duo.
So acids are proton donors.
Yep.
And bases are proton acceptors.
You got it.
It's like a constant give and take with those protons or H+ ions being exchanged, right?
Exactly, like a molecular dance.
And we measure this with the pH scale, don't we?
I do.
So the pH scale measures the concentration of those hydrogen ions in a solution. But here's what I find so interesting. It's a logarithmic scale.
Yeah, that can throw people off.
It can.
Yeah.
But it means that a change of 1 pH unit represents a 10-fold change in hydrogen ion concentration.
So a small change on the scale means a big difference in acidity or alkalinity.
Exactly. A solution with a pH of 3 is 10 times more acidic than a solution with a pH of 4.
And 100 times more acidic than one with a pH of 5.
That's what I always emphasize to my students, that logarithmic nature can be tricky. But it's so essential to understanding those dramatic effects even small pH changes can have on those biological systems.
They can really mess things up.
Totally.
Yeah.
And it explains why keeping a stable pH within such a narrow range is just crucial for survival.
Yeah, think about it. Our cells are always producing metabolic acids, lactic acid from exercise, ketone body sometimes. If our pH fluctuated a lot, all those critical biochemical reactions would just stop.
They'd just grind to a halt.
Exactly.
Okay, so how do our bodies maintain that super delicate pH balance then?
Well, I bet you know what I'm going to say.
Buffers.
Buffers. They're the heroes of pH regulation.
Okay, break it down for us.
Okay, so buffers, they're mixtures of a weak acid and its conjugate base. They're like chemical sponges, soaking up extra protons when things get too acidic, or releasing protons when it gets too basic.
So they help to resist those huge pH swings.
Right.
They could cause chaos in our cells.
Exactly. And this is where that Henderson-Hasselbalch equation comes in.
Oh, yeah, the one we talked about in class.
It's not just a concept, It's a tool that helps us understand how buffers work.
Okay.
And how well they'll resist those changes in pH.
So it relates the pH of a solution to the pKa of the buffer.
Which is that negative log of the acid dissociation constant, by the way.
Right. Right. And the ratio of the concentrations of the conjugate base and the weak acid.
Exactly.
So if we know the pKa of a buffer and we know what pH we want, we can use that equation to figure out the best ratio of conjugate base to acid.
Absolutely.
To get the best buffer for that situation.
You got it. And that's important in the lab, but also for doctors.
Oh, right.
They use that Henderson-Hasselbalch equation to understand and manage acid-base problems in patients.
Right.
Like those with diabetic ketoacidosis, again.
Okay, let's talk about some specific buffer systems that are keeping our bodies going.
What is this?
So the main buffer in our extracellular fluid is the bicarbonate carbonic acid buffer system.
That's the big one.
And that's a dynamic interplay between carbon dioxide, carbonic acid, and bicarbonate.
All kept in check by carbonic anhydrase, too.
Oh, that's that enzyme we were talking about earlier, right?
That's the one.
And it's found in red blood cells, which makes sense because red blood cells aren't just transporting oxygen.
No, they're not.
They're playing a big part in pH regulation, too.
They are busy little cells.
Yeah. So they use both bicarbonate and hemoglobin to buffer blood pH.
Right.
And hemoglobin, it can bind to both oxygen and protons, making it key for gas transport and pH regulation.
A true multitasker.
It's amazing how these systems all work together so smoothly.
It is.
And, of course, we can't forget about the respiratory system and the kidneys.
Oh, no, they're vital.
So important for pH balance, right? The respiratory system controls CO2 levels through breathing.
Yep.
While the kidneys, they excrete acid.
And reabsorb bicarbonate.
Right. And this back and forth between the respiratory and renal systems, it's so crucial for keeping our blood pH within that narrow range.
That narrow, life-sustaining range.
Okay, we've talked about those big players in extracellular pH regulation. But what about inside the cells? Are there other buffer systems there?
There are.
To keep things stable?
Absolutely.
Yeah.
So phosphate and proteins, they're the main intracellular buffers.
So phosphate ions, particularly dihydrogen phosphate, H2PO4, and hydrogen phosphate, HPO42, they can accept or donate protons.
That's right.
And proteins with all their amino acid side chains.
Right.
They can act as buffers, too.
Yep. Some of those amino acid side chains, like histidine, they have pKa values really close to physiological pH.
Right.
So they're great at buffering.
It's a multi-layered defense system.
It is.
With those buffers working on both the outside and the inside of the cell.
Absolutely.
To keep the pH in check.
That's right.
Now let's talk about a specific acid in the body. Hydrochloric acid, HCl, in the stomach.
Ah, yes. That's a powerful one.
Incredibly strong. Essential for protein digestion.
Uh-huh.
But it could be damaging if it wasn't controlled.
Right. Oh, yeah, for sure.
So how does the body handle that?
So that's where the small intestine comes in.
Okay.
It secretes bicarbonate to neutralize that acidic chyme from the stomach.
So it's protecting the lining of the small intestine.
Exactly.
It's fascinating how our bodies can use acids, but also protect themselves from them at the same time.
It is a really delicate balance, isn't it?
It is. So now it's time to put all this knowledge to the test.
Ooh, are you ready for some Q&A?
I think so. Let's see if our listeners have been paying attention.
Okay, first question. All right. Future biochemists, are you ready for round 1 of Q&A? Let's see if you've been paying attention to those lectures.
I'm ready when you are. Let's start with a question that often trips students up. How does that dipolar nature of water make it such a good solvent? Think back to those hydrogen bonds and how water interacts with other molecules.
All right, so remember, a water molecule has a slightly positive end and a slightly negative end.
Right.
Because of that unequal sharing of electrons between the oxygen and hydrogen atoms.
Right.
That's what lets it form hydrogen bonds with other polar molecules and ions.
Okay.
So these attractions, they effectively surround and dissolve the solute.
Making water a great solvent.
Exactly.
Okay, next up. What's the key difference between a strong acid and a weak acid? Think about how they dissociate in solution.
Think back to those demonstrations we did in the lab. A strong acid, it completely dissociates in solution, releasing all its H+ ions. A weak acid, though, it only partially dissociates, and its tendency to donate protons.
Yeah.
That's reflected in its Ka value.
Okay.
The higher the Ka, the stronger the acid.
Gotcha. Now let's tackle the Henderson-Hasselbalch equation.
Oh, yeah, that one.
I know lots of students find it intimidating, but it's so important for understanding buffer systems.
It is.
So how does it help us understand how those buffers work?
It's all about relationships. The Henderson-Hasselbalch equation, it connects the pH of a solution, the pK of the buffer, and that ratio of the conjugate base to acid.
Okay.
It helps us predict how a buffer will respond when the pH changes.
Right.
And to figure out the optimal pH range for a specific buffer system.
It's a must-know for the exam. Okay, moving on. What is the main buffer system in our extracellular fluid? Think about the role of carbon dioxide in red blood cells.
That would be the bicarbonate carbonic acid buffer system.
That's the one.
It's a dynamic equilibrium involving the interconversion of carbon dioxide, carbonic acid, and bicarbonate.
But we can't forget about carbonic anhydrase.
No, absolutely not. That enzyme is key in this system.
It is. Now, how do those hardworking red blood cells contribute to both pH buffering and CO2 transport? They're multitasking marvels, aren't they?
They are. They use both bicarbonate and hemoglobin to buffer that blood pH and transport CO2 to the lungs. Hemoglobin is a real workhorse, you know?
It is.
It can bind to both H+ ions and CO2, helping regulate pH and remove metabolic waste.
Okay, switching gears a bit. What are the main intracellular buffer systems? Think about what molecules are abundant inside those cells.
Remember those phosphate groups? And all those proteins with their diverse side chains?
Yes.
Those are our main intracellular buffer systems, phosphate and proteins.
So those phosphate ions, particularly H2PO4 and HPO42, they can accept or donate protons.
Right. And proteins, with their various amino acid side chains, can also be effective buffers.
It's like a tag team effort.
It is.
All right. Onto those kiddies now. They're not just filtering waste. They're playing a big role in pH regulation, too.
They are multitaskers as well.
How do they contribute?
Well, they fine-tune the pH by excreting acid, mostly as ammonium ions, NH4+, while returning bicarbonate back to the blood.
To keep everything in balance.
Exactly, to maintain that overall acid-base balance.
Okay, last question for this round. What makes hydrochloric acid so unique in the body? Think about where it's found and the challenges it brings.
Hydrochloric acid is unique because it's so strong, essential for digesting proteins in the stomach.
Right.
But potentially damaging to other tissues.
It's a double-edged sword.
It is. But the small intestine neutralizes it.
With bicarbonate.
Right. That prevents damage to the delicate intestinal lining.
It's really amazing how our bodies can harness the power of acids.
It is.
But also protect themselves from them.
Delicate balance indeed.
Okay, we've been focusing on water, acids, bases, and buffers.
The fundamentals.
Yeah, but those are really the foundation for understanding so much more in biochemistry.
They are.